It's a great pleasure to be here and presenting our recent work on a computational framework to model conformational changes of proteins upon interactions. And in our lab, with the ultimate goal of understanding and modulating cell behavior, we focus on algorithm development at the molecular level. And the the molecule of particular interest is protein. And with the technological advancements over the year, we are accumulating a quickly growing list of protein sequences with unprecedented speed. In the meantime, the number of proteins that have been structurally determined or functionally annotated remains flat, so there's a huge gap to fill. Not only for individual proteins, for protein-protein interactions, if we look at even a small E. coli interactome with thousands of validated protein-protein interactions, only less than 10% have structural information, and this limits our capabilities to modulate cellular processes as well as our insights underlying into these cellular processes. So we believe that computational methods, for instance, protein docking, can help fill the gap. It starts with individual protein sequences and structures, and it can help predict the structures of the bound complex. And this can be combined with relatively low-resolution partial information from experiments. So this problem can be solved computationally thanks to the free energy minimum principle. From the perspective of free energy minimization, there are three factors. Each factor actually presents its unique computational challenge. The free energy models are often approximate, costly to evaluate, and extremely rugged, but we are getting more and more help from statistical potential, experimental data, and coevolution data. And the search space consists of six-dimensional, relatively rigid body motion, and it's actually a Riemannian manifold with curvatures, and it's really hard to optimize upon. Not to mention that there are additional high-dimensional internal flexibility conformational space. And given this background, it's extremely hard to find efficient and accurate search algorithms. Over the years, we made contributions, first reparametrizing the six-dimensional rigid body space by using the by using the exponential coordinates, sort of flattening the manifold, make it really easy to optimize. And then we devised an optimization method in this reparametrized space, which is tailored to the free energy landscape, which is the protein docking funnel concept. And more recently, we focus on the additional internal flexibility. In this talk, I'll talk about dimensionality reduction for this high-dimensional internal flexibility space with physics-based models, that is normal mode analysis. So if we talk about protein flexibility and how it's treated in protein-protein interactions, it's really a broad spectrum. Each corresponds to certain mechanistic concept about protein-protein interactions. Starting from the far left, we treat proteins as just rigid, and then protein-protein interactions are just key and lock. And this has been proven to be unrealistic, but if we go all the way to the right, it becomes computationally intangible. So somehow we have to find a sweet spot in between. And the sweet spot we found in this work is so-called normal mode analysis. Here you represent each residue or heavy atom as a node, and then you put harmonic potentials on the edges between neighboring nodes, and then you can calculate the Hessian and eigenvectors of the Hessian, and these eigenvectors are normal modes, which turn out to capture the protein intrinsic flexibility very well, especially those flexibility motions exhibited in molecular dynamics or NMR ensemble. So the question for us is intrinsic flexibility observed in normal mode analysis enough to capture conformational changes in protein interactions? Well, the answer is yes, if you believe in conformational selection model that is saying that proteins pre-exist in an ensemble of unbound conformations, and the bound conformation is actually included. When it's interacting with another protein, the bound conformation is selected for. So if you believe that this is adequate, you would think that the intrinsic flexibility should capture the conformational change very well. Actually, there are two seminal work which tells that it's pretty much the case. If you look at the normal modes calculated on the unbound proteins, actually this normal modes correlate with the observed structural differences or the conformational changes, but there's always a catch that they found there are some cases where conformational selection alone is not adequate. So what do we search beyond intrinsic flexibility? So beyond the structural conformational changes elicited from intrinsic flexibility, we think there might be also induced conformational changes by its binding partner through intermolecular interactions. And we believe that both models play a role, and why don't we consider both in one framework? Considering that normal mode analysis has already done very well for the left arrow, we decided to expand the normal mode analysis for this purpose. So in today's talk, next I'll quickly talk about how we extend the potential model and how we found the peculiarity about the Hessian and normal modes, and how we extract modes useful for anticipating conformational changes, and lastly, empirical results and mechanistic insights over a large-scale test set. So as I said before, if we just look at individual proteins, we can have nodes and harmonic potentials over neighboring nodes. And but when we were talking about proteins, they come to each other and they form contacts and form an encounter complex. And this complex is transient, more or less, and depending on how tight it is, the proteins dissociate and then form another encounter complexes. So if we put the protein flexibility in the context of encounter complexes, we have two individual anisotropic network model capturing the intermolecular potentials. And then beyond that, we need to introduce the intermolecular interactions. As a stepping stone, we decide to use intermolecular potentials in a similar fashion, which is also harmonic potential like the intramolecular potentials, but we differentiate the distance cutoff as well as the stiffness of the springs. So these are the two parameters we are going to vary in our experiments. So how does the Hessian look like? It turns out that the Hessian actually has a very peculiar structure. It has a dimension for protein 1, let's call it a receptor, and dimension for protein 2, let's call it a ligand. So the off-diagonal blocks that correspond to the coupling between the receptor and the ligand. It's a very sparse matrix. The elements are 0 except for those pairs which correspond to intermolecular interactions. Then if you look at the diagonal matrices, they are actually resembling the Hessians you will get for individual proteins, receptor or ligand, in the conventional normal mode analysis. But the tilde here represents that there is a slight difference. And what that difference looks like. In fact, if we look at the Hessian, the previous one on the diagonal, we have the Hessian matrix corresponding for the receptor. And if we have m nodes or atoms, there are essentially m square superelements. Each corresponds to the coupling between atom I and atom J. And each superelement is a 3 by 3 matrix because either atom has 3 degrees of atom. So it turns out that compared to the corresponding elements that you get from the conventional normal mode analysis, only diagonal superelements can differ, and these superelements are corresponding to those atoms that can form intermolecular interactions. In other words, only the different degrees of freedom of the same atom which forms intermolecular interactions, the coupling has been changed. And if we look at a particular element, we can see that the difference comes from an additive term due to different interactions with its binding partner. So given this peculiar structure of the diagonal block, we wonder whether this matrix actually captures the intrinsic flexibility but modified by some induced fit effects. So the first try is to actually utilize this matrix. We had to do some projection because of the basic assumption of normal mode analysis I won't have time to talk about, but then we calculate the eigensystem and look at the eigenvectors and ask how good are these normal modes. And we test them on a test set which is popular for protein-protein docking, and it includes over 170 pairs of proteins, and they hover non-redundant sequences and the structural folds, and they have various extensive conformational changes. And you may ask, how do you find an encounter complex when your ultimate goal of protein docking is to put two proteins together? We cannot experimentally observe encounter complexes easily, but we can approximate them by docking unbound proteins rigidly. So we have two scenarios. In the first, you just geometrically fit individual unbound proteins to bound. This captures sort of the optimal scenario you can get from unbound rigid protein docking. And in the second case, which is more realistic, you don't use any information from the bound complex, that you just do energetically docking and you pick the top 10 predictions. And we focus on the cases where the conformational changes are significant, above 2 angstrom. That corresponds to 113 individual proteins, either in one geometrically fit encounter complex or 10 different energetically docked complexes. So how do we assess how good are the normal modes? Okay, so if between unbound and bound proteins, the difference is usually captured by the so-called RMSD. So starting from the unbound structure, if I deform the unbound structure along a linear combination of the top K normal modes, how close can I get to the bound? So this optimal solution actually has an analytical expression like this. So if you have this, you can explicitly calculate the optimal RMSD reduction. So this is the measure for either method for each case. Now, how do we compare the two methods over all cases? We introduced the two metrics. One is average improvement and one is population improvement. The average is just comparing the two RMSD reduction and averaged over all cases. So 0 indicates a tied performance. But to prevent the cases where few extremely good or extremely bad cases dominate the average performance, we also look at the population improvement. We ask what's the percentage of at least 5% improvement in RMSD reduction compared to the conventional normal mode analysis, and subtracted by the at least 5% deterioration. So here, this is sort of a net success. We actually penalize the mediocre performance where the two methods don't differentiate by more than 5%. So again, 0 indicates a tied performance. So how does the results look like? On the left and on the right, we call this the submatrix method. On the left, you have the average results, and on the right, you have the population results. So without the burden of asking you to look at different curves corresponding to different parameters, and then on the x-axis, we are increasing the number of top modes. If you just look at this dashed straight line, which is 0, you will find that except for the first few normal modes, our curves are all below 0. That indicates that our performance is worse. So this is, of course, a hard conclusion to accept, but a silver lining was that if you look at this dashed line, which corresponds to the cases where you don't differentiate inter- and intermolecular interactions, actually the performance is worst. In the other words, if you just blindly apply normal mode analysis, conventional normal mode analysis to an encounter complex, the performance will be very bad. So, but how do we move on? Let's go back to the Hessian matrix. What did we use was just the coupling, the induced motion of the receptor or the ligand, but we sort of disregarded the induced motions of its binding partner as well as a coupling between the two sets of motions. So why don't we just use a whole complex-derived normal modes and see how it performs? So this is a new performance. Apparently, the results are much, much better, many curves above 0, and the net success rate can be even more than 30%. And an interesting factor looking at the different parameters is that the intermolecular potentials, the stiffness parameter has to be smaller. It's a quarter compared to 1, which is satisfying because we know non-bonded interactions dominating intermolecular interactions are generally weaker than the chained connected bonded interactions. And the distance cutoff turns out to be that it has to be smaller than the intermolecular distance cutoff, like 12 versus 15, but it cannot be too small. So it has to be beyond 6 angstrom or the first layer interactions. So now that we have good results, how do we, and also, the last one was for the geometrical fit complex, and this is for the docked complexes. Actually, in this realistic cases, we are doing even better than the conventional normal modes. As you can see, the curves are all moving up. So now that we have good results, how do we know that they are not better for wrong reasons, right? For us, we are particularly interested in was intermolecular interactions the sole factor behind the improved performance. And the equivalent question to ask is, had these intermolecular interactions been removed, would the new method reproduce the conventional normal mode analysis? A quick answer is yes, but only after re-ranking. And not a so quick answer is, if you look at the Hessian, the complex derived Hessian, and then now we have no intermolecular interactions, so the off-diagonal becomes 0, and the diagonal ones become just like the conventional Hessian you observed. And this matrix actually has peculiar eigenvectors and eigenvalues. So the eigenvalues or eigenvectors of individual diagonal components are like this. But then you ask what the eigenvalues and eigenvectors of this big matrix now looks like. It turns out that it's just the mixed eigenvalues from the two individual block matrices, and the eigenvectors are again just remixed by augmenting the other dimensions. So it's like remixing two songs. So in other words, it's totally entirely possible that sometimes the top modes of this complex derived Hessian can be dominated by either receptor or ligand. It turned out that it is a case that if we have interactions and ask for individual protein complexes on each row and ask its number 1 through number 6 eigenvectors, who do you resemble? Who do you resemble, ligand or receptor? The red would be a receptor and the blue would be ligand. Some are dominated by receptor and some are by ligand. So indeed, we have to use the rescaling approach. I won't spend too much time on it. And after rescaling, we could reproduce the conventional normal mode analysis. So how does the sub-vector approach after rescaling works? It turns out that it's not as good, but it's still above 0, indicating that the maintained improvements, again in normal mode analysis, proved that the sole contribution from the intermolecular interactions, because now we know that there cannot be other factors. And another interesting factor is that the bands of curves are coming closer to each other, indicating improved sensitivity to parameters. And the last question we sort of ask is, you had better results, but how do you know that you're not benefiting from extremely good encounter complexes? Or in other words, how good do these encounter complexes have to be? If we look at from left to the right, the quality of encounter complex goes from good, bad to ugly. And then for all such encounter complexes, we ask in each bin, what is the optimal RMSD reduction, or what is the predictive power? The higher, the better. So we sort of see that if we look at the frontier, or we just sharpening this cloud into a curve by averaging in our mind, we see a slightly curve like this, which indicates that better encounter complex does help, but it's not that sharp turning. In fact, you can still have good predictive power or RMSD reduction with sort of mediocre 20 angstrom models, which are often classified as incorrect models in Capri. So lastly, I'd like to summarize that so far we used mechanistic concepts to inform our algorithm development. So we used both contributions of conformation selection and induced fit in the same framework of normal mode analysis, and we reduced the high-dimensional conformational change into just a few predictive collective motions. And it provides a dimensionality reduction tool for protein docking. So we can use your favorite protein docking free energy models. You can use your favorite search algorithm, and you can do so in the reduced flexibility space for better docking results. And the performance of the cNMA really provides further mechanistic insights into protein-protein interactions. So we not only supported that both contributions play a role, we also showed that not only induced motions of a protein under study, but also those of its binding partner, and the coupling between the two sets of motions led to the improvements. And at the end, I'd like to thank, this work is much done by my postdoc, Tomasz Aliwa, and the docked encounter complexes are provided by the Jiping Weng and Tom Warren from UMass Medical School. And we built our software on top of an open source program by Yvette Bahar and Ahmed Bakan. And at University of Pittsburgh, this project is funded by National Science Foundation as well as startup from Texas A&M. And we are really recruiting PhD students and postdocs to study protein interactions, the protein small molecule interactions. We're particularly interested in understanding and overcoming drug resistance mutations in infectious disease and cancer treatment. And with that, I'd like to thank you for your attention and I'm happy to answer any questions.
Two quick questions. The first, how intensive it is in terms of computational requirements, your method?
Actually, computational-wise, is not that extensive at all because we are looking at the C-alpha level normal mode analysis. So calculating the Hessian or the normal modes are extremely easy. The computationally costly part is actually for our analysis, trying to fit the normal modes through the conformational changes. That's just for posterior analysis.
Are you planning to adapt your method to protein-DNA interactions?
Pardon me?
Are you planning to adapt your method to protein and DNA
Absolutely.
interactions?
So as I said, we previously focused on the other factors, but here we really want to isolate the single factor of the conformational space. So we believe that with the guidance of a good free energy model and an efficient search algorithm, you will be able to improve your protein docking methods in this reduced but still very informative subspace of conformational space.
Okay. Thank you very much, Shen.
Thank you.